Hey, let's start our webinar. Good evening.
Good evening.
It gives me great pleasure to warmly welcome all of you to this special session jointly organized by the Young Scientist Forum, National Science and Technology Commission, and Ecosphere Resilience Research Center, University of Sri Jayewardenepura. Today's webinar session will provide valuable insights into sampling and analytical techniques for detecting microplastics in water. So we are very confident that this session will offer you valuable insights and impactful takeaways. Now, let me introduce our esteemed resource person for today's event, Dr. Kushani Mahatantila. Dr. Kushani Mahatantila is an environmental scientist from Sri Lanka, currently serving as a senior research scientist at the Industrial Technology Institute with a PhD in environmental science from Shimane University, Japan, and expertise in water quality analysis, environmental pollution remediation, and ISO IEC 17025 laboratory standards. She has contributed many significantly to national and international environmental projects. Her research focuses on freshwater systems, microplastics, and heavy metal contamination. Her work exemplifies a commitment to advancement in environmental science and sustainable practices. Dr. Kushani, we are truly honored to have you with us, and I encourage all of the participants to take this opportunity to engage actively during the session. Over to you, Dr. Kushani. Thank you.
Thank you, Dr. Anushka. So I'm planning to talk about the sampling and analytical techniques in microplastic detection in water systems. So what do you think? What are these particles? The red particles, yellow ones, green, the blue particles. What are those particles? Actually, those are particles we collected last year from Canyon River. These are fibers, foam, sheets, and fragmented particles. They are all microplastic particles. So, in different publications, microplastic defined in various ways. However, I took this definition from the ISO standards ISO 24187 and ISO 21960. They define microplastics and large microplastic as any solid particle insoluble in water with any dimensions between 1 micrometer to 1000 micrometers as microplastic. Typically, a microplastic object represents the primary intentionally added to end-user product, such as cosmetic means and coatings, paintings. Those are primary microplastic, and the secondary microplastic objects can also result as a fragment of the respective item. And also, the plastic may show various shapes, and the defined dimension is related to the longest distance of the particles, where large microplastic are any solid particle insoluble in water with any dimensions between 1 millimeter to 5 millimeter. However, in the recent standards, which is to be published early next year, it encompasses both definition into one and define only microplastics as any solid plastic or synthetic polymer particles insoluble in water with the largest dimensions between 1 micron to 5 micron, and the term microplastic covers the sum of several individual microplastic particles. So why we especially talk about the freshwater environmental and the detection of microplastic in water? Because water systems are easily vulnerable to the pollutions. As we all know, the rivers like Kalani, Kaluganga, and all our rivers contaminated with microplastics through urban runoff, untreated sewage and wastewater disposal, and agricultural drainage. Without no doubt, our marine environments are also contaminated highly with microplastics. Recent studies also indicated the groundwater contaminated with microplastic near landfills and agricultural zones. These microplastic particles are very durable, so they can percolate through the soil and sediment systems and posing risk to the potable water, because we all know we take fresh water and groundwater for drinking. This illustration shows you the water contamination and enter the microplastic into the food chains and humans. The wear and tear particles from the tire, from road runoff, the degradation of plastic litters, disposal of wastewaters into rivers adds microplastic to the water systems, as well as washings, because we use a lot of synthetic clothes, and those clothes contribute to a lot of microplastic also. When you use this face scrub, toothpaste, the tiny beads for exfoliation are of microplastics. They enter in the water systems, and the aquatic organisms from algae to the large fish consumes those microplastic, and through the food chains we get. And even some of the microplastic enter to the air systems through inhalation and ingestion. In both ways, we get microplastic into our bodies. So when we talk about the detection techniques of microplastics, the detection is mainly from the collection, sample preparation, and identification of microplastics. When we search the research from early in 2019, the most of collection of the microplastic nets are mostly used, then pumps, and then sieves. Some of the studies has used 25% of the studies has used density separation for separate the inorganic particles, but 75 research has not been used any density separation process. For the digestion, there are several solutions are used, like hydrogen peroxide, enzymes, alkali digestion, and acid digestions. 60% of the researchers has not used any digestion, and 25% has used hydrogen peroxide as oxidative digestion. Identification has been done by visual methods, FTIR, Raman, scanning electron microscopy, and dyeing methods. So 40% are visual methods and 40% of them are FTIR methods. However, this is a new field, and as the comparison, we know we don't have an exact standard method to analyze the microplastics. Still, the ISO standards are being developing. They are the microplastic in water, analysis of microplastic in water standards due to be published next year. So it is very difficult to compare the results. We know for the regulations, we need the baseline data, and to compare the baseline data, the research has not been done in a systematic way. They have used different methods, different quality control measures, so the data cannot be compared. There are some replication issues and quality control issues as well. So there's a huge need of standards method for the microplastic analysis in water systems or any other matrix. So this is the schematic representation of interdependencies during microplastic analysis. So the sampling method specifically depends on the sample being analyzed. and the sampling volume depend on the detection techniques. If we want to sample environmental sample like fresh water, we can use the nets like Manta or Newton and sieving methods. For the groundwater, we have to use pumps, or sieving can be in situ sieving can be employed for the groundwater sampling. However, if we are analyzing the drinking water in the bottled drinking water, so we have to sample a product from the representative number of the water bottles according to their production. However, the detection techniques depend on the target group with evaluation concept. If we want to analyze the ecotoxicology, then number of particles, size of particle, distribution of particles and shapes are important, because the smaller the size that can be entered to our body through the digestion tracts. And the polymer type is also important. But then we have to use spectroscopic techniques. From the gas chromatographic techniques, we can get a mass fraction and polymer types, but not the particle size or the particle number. So like that, depending on the objective of our research, the detection technique is varied based on the detection technique. Sample volume should be decided, and preparation of samples should be based on the detection techniques. This is the interdependencies of microplastic analysis: the sampling, preparation, and detections. In environmental samples like fresh water, we can, as we discussed earlier, the most common used method was using the nets. There are Newton nets and the Manta nets. The difference is the Manta net contain a buoyant wings. So when we employ the Manta nets into the water, we also incorporate a flow meter. So the flow meter rotates according to the flow rates with the number of rotations into flow meter constant. We can calculate, and the area of the mouth of the net, the area of the mouth of the net submerged in water, we can calculate the volume of water being sampled. It is easy to use and collect large volumes of water. In Kalani River instance, for a half an hour period, we collect minimum of 6 cubic meters of volume of samples, 6 cubic meters. It is widely used, but it produces large number of microplastic into net. But the Manta net is expensive, and there's a limitations. The net mesh size is nearly 300 microns, so we could collect only the larger particles than 300 micrometers. It needs boats or stepped into water. And plankton nets. The main advantage of the plankton nets is the lower pore size of the net. It limits up to 100 micrometers that can collect more particles. However, the sample volume is less than the Manta and Newton nets. This is how we collected water from Kalani River using Manta nets. In the upstreams, we use step-on system. We install a metal rod, and from that we use the Manta net and keep for 30 minutes. So after the 30 minutes, we take the Manta out and wash the net to collect all the particles in the cod, and then with high pressure flow of RO water, we transfer the particles into glass bottles. Then it is covered with alumina foils and capped with steel lids. While transferring the Manta net sample to our container, our container, second jar was opened and closed simultaneously with the sample jar to estimate the air deposition during transferring the water sample. Because if there's a lot of particles in the air while transferring, they do not represent the actual microplastic in water sample. So we did an air blank when transferring the water into our sampling jar. So we did a blank testing for the high pressure washer and stored the sample at 4 Celsius until analyzed to reduce the microbial degradation further. In standard, for the principle for analysis of microplastic present in the environment, which I mentioned 24187, it also recommends the storage at 4 Celsius until analyzed to reduce the microbial degradation of the particles further. Other than the nets, the sieving and pumps are available. That the sieving, the advantages does not require specific equipment or boat. We can use the cascade of sieves and ease to collect the samples and allows choices of mesh sizes. I will talk the mesh sizes in next slide. But this is laborious and time consuming. Sample medium volume of water and we have to transfer the water with buckets. However, when we want to sample a water column, we cannot use the sieves or nets. Then the only option is pumps. For the particular depth we want, we can employ the pump and sample the water. In there's advanced, it allows the choice of different mesh sizes. As I discussed earlier, sample volume depends on the detection limit of the selected analytical techniques. And in the standard 24187, fractionated filtration is recommended to reduce the blockage of filters. That is, you can use different different mesh size sieves to filter the water. The mesh size can be selected according to their particle size classification. However, during the filtering process, recovery tests are recommended to assure the sample losses or addition during the filtering and transferring. No recommendation for preferred sampling, means of random sample or aggregate samples to be collected. Still, there's no such a recommendation. As I mentioned earlier, this is the particle size clarification. So if we use the fractionated sieves, for the first sieve, we can use 5 millimeter sieve. So particles retained on that sieve, we can consider they are macroplastic, and if we want, we can analysis. For if we are interested only on microplastic, we can discard the particles retained on the 5 millimeter sieve. Then we can fit the 1000 micrometer or 1 millimeter sieve bottom to the 5 millimeter sieve. Then particle retained on the 1 millimeter sieve, we can collect them as large microplastic. And then we can use the 500 micron sieve, and it gives these fractions. Like that, according to this, the standard which are going to introduce from next year, they use the same particle size classifications for the sieves and everything, and they recommend this particle size distribution to be considered by sampling and analyzing with detecting the samples. However, you can see when we consider the mass of different particle sizes, here, very low masses, when the number of particles which gives 14.13 milligrams in the large microplastic particle, this achieved by one particle, and once the average particle size reduced to 300, that is reduced by 10-fold, the number of particles increased by 1,000-folds. That is why when we analyzing the mass fraction, we could have higher number of samples of smaller particles. So to achieve the LOD, it may need lot of particles if the sample size is very small. So it is challenging for the mass fraction if we are analyze based on the mass fractions. For the removal of inorganic residues, once we collected into the nets and every nets, we have the inorganic fractions, that is sands and so on. So that is separated by density separation. We use solutions of different densities to separate. Less dense materials floats and separate from the denser sinking materials. So that saturated sodium chloride, the density is 1.2 grams per cubic centimeter. So using the sodium chloride, we can separate polyurethane up to polyurethane only. But when we use the zinc bromide or zinc chloride, the density is 1.6. Zinc bromide density is 1.7. If we use these compounds, we can separate up to POM. However, that sodium chloride is less toxic to environment than zinc chloride and zinc bromide. So when employing the methods, we have to consider about our objectives as well as the environmental pollution also. So after removing the inorganic fractions, the organic fractions sometimes extracted with the microplastic because they also less dense. So removing the natural organic matrix are proposed for all the analytical dyeing spectroscopic techniques and in specific cases for thermal analytical methods like pyrolysis GCMS. The treatment with oxidation solutions, the phantom regions are most frequently proposed and reported as the best oxidizing solution. However, there are several advantages and disadvantages of different solutions. Acid digestion is normally, it is fast, but it digests the particles of interest, so it is not recommended. Alkaline digestion and oxidative digestions are recommended. Enzymatic digestion, the effect to the microplastic particles are less, but it requires the to control the temperature. Enzymes are very sensitive, so in temperature conditions are enzymes are very sensitive to the temperature, so we have to carefully control the temperature if we are using the digestion. But specifically, it is specific to some compounds. So some natural organic matter may not be digested with the enzymatic digestion, and enzymatic digestion are very expensive. However, in my laboratory, that for the sampling of environmental waters, as I shown earlier, I use manta troughs, and the drinking water, I take the bottles from the production, at least 3 bottles from one batch, and do the visual inspections. This is the method I used. Visual inspection, if the low organic matter content is present and clear water like drinking water, for the 400 milliliter of the sample, I add 30% KOH and NOCl solution and incubate for 24 hours at 40 Celsius with magnetic stirrers, which are glass, not plastic, and keep in the shaker incubator for 24 hours or the incubation that stirrer, temperature control stirrer for 24 hours. Then filtered through the glass fiber filters. That GFD filter has the pore size of 2.7 micron, so I can collect the particles greater than 2.7 microns in diameter. Then I use the staining method for the particle staining and staining with Nile red, and images were taken under blue light with orange filters. That is, I use certain energy to get the fluorescence from the stained particles. If the high organic matter or high suspended solids are present, I do everything up to here, and only the supernatant is filtered first, and the sediment deposited is wet sieved using 350 micron sieve and collect content in a clean beaker and again filter. If the filters are clogged, I use several filters and the staining is carried out. This is my sample preparation when I do at the laboratories. That we work with the CFAS UK, and this method was developed by them, and in the laboratory network, they use the same method, same methods, same net sizes to compare the results. So as far as possible, within several countries, they have employed laboratories and used the same method in order to compare the results. So this is the method, and this method gives 80% recovery. So when we consider the detection methods, the first microplastic analysis in 2004 reports the visual identification under stereos microscope or optical microscope. When the particles cannot be identified accurately, then the researchers have used the hot needle that is around 130 degrees Celsius and see whether the particles melt or not. That is how they identify the plastic. But which time the detection methods had developed very fast, and there are so many sophisticated methods available now. This is the Nile red method, and I mentioned earlier the fluorescence particles here are microplastics. Some researchers has used scanning electron microscopic images for analyze the microplastic, and it is very few researchers has used ICP-MS method to analyze particles from microplastic to nanoplastic particles. That with normal ICP-MS, we cannot analyze the nanoparticles up to submicron particles. It needs special nebulizers and high sensitive ICP-MSs. The particle analyzers for ICP-MS, that method is called single particle ICP-MS analysis. They are like silver nanoparticles can be easily analyzed, or in some researchers, they have coated microplastic with the gold particles, and those particles are analyzed. With that analysis, they can count the signals. One signal is given by one particle, so they can analyze the signals and count the number of particles, and with the single signal intensity, they can analyze the quantify the amount. And also the polystyrene particles coated by sulfur has been analyzed by triple quad ICP-MSs. And as spectroscopic method, micro FTIR and micro Raman spectroscopic methods has been developed to analyze microplastic, and pyrolysis GCMS, which means there's a pyrolysis column to melt and volatilize the particle, And in the pyrolysis, the plastic particle is break down into decomposition byproducts, which is smaller and more volatile than the original compound. Then they goes through the GC column and separated and analyzed by mass detector. These are the commonly available detection limits used by researchers for the analysis of microplastics. And in 2025, the ISO methods of water analysis will address the micro FTIR, micro Raman, and pyrolysis GCMS methods. What are these particles? Actually, these are blank contaminations because the standard says the microplastic should be analyzed in ISO class 3 rooms, which is not affordable for us. That is, the lab should have a laminar flow systems, dust collector system that the laboratory should be free of dust, and we cannot go for such a laboratory. It's like pharmaceutical testing and pharmaceutical manufacturing and semiconductor testing laboratory facilities are recommended for the analysis of microplastic because our environment is contaminated with microplastic. It is difficult to maintain unpolluted preference or air blank sample with microplastic. Those are the particles reported by one researcher. They are black contaminations and air contamination. Actual sense in my laboratory, we also do a air blank during our samples. Normally, when the analysts are using synthetic lab coats, for a period of 1 hour, we can collect like 3-4 particles for our samplers. So how can we minimize the contamination? How can we reduce the contaminations? Instead of synthetic lab coats, we highly recommend cotton lab coats when analyzing the microplastic. Hand wash and no gloves are recommended, but if you want, we can use only nitrile gloves because they are highly resistant to chemicals than the other gloves. For the sample preparation, we can use the laminar flow environment. Just laminar flow for the sample preparation, sample weighing, and then the working for the conditions. And we can clean the glassware properly with RO water, and when not in use, we can cover with aluminum foils to contaminate with dust. And all the filters, we have to wash the filters, and we have to do a blank for the filters also. No masks are recommended. If you want to wear a mask, only N95 masks are recommended. And it is recommended not to use makeup, creams, toothpaste, and shampoo containing microplastics. And also the loosened hair is not recommended. The filter papers and the sample should be protected in the glass containers or the steel containers. That if we are the samples, if we are using the bottles and everything, it is recommended to external cleaning before the sample opens. So to minimize the environmental contamination. So as I discussed earlier, method of detection, it was microscopic identification. When the particles are larger than 500 microns, then we can mount it onto the FTIR and identify the polymer type. Otherwise, visually, we can identify the number of particles, shapes, color, and everything. And also the size. For the measurement of size, several softwares are available now. And this is the 9-red method I employed in my laboratory. Once we stain the filter paper, we mounted it on the engraver machine, and we take under blue light, we take under blue light and orange filters, the microplastic particles gives fluorescence, and those fluorescence particles can be counted manually or automated system. But when we are using an automated system, we have to validate that system with the different color intensities. So validate the systems, it is recommended to add the known amount of non-polymer type particles and see whether they are to validate the system, we have to, after adding the known amount of particles of non-polymers, we have to calculate the recoveries by after adjusting the different colors. So this is how the filter paper looks in white light, and this is under blue light. These fluorescence particles are counted manually or automated, computer-aided softwares. And by visually or under microscope, we can see the shapes and color of the particles. This is the micro Raman spectroscopy, but the technique what we use here, the Raman spectroscopy normally use the laser light source, and from 400 to 2000 microns, nanometer, the wavelengths. And the Raman shift is that when we give the light energy to the particles, that the electrons, outer shell electrons are excited, and once they shifted in the ground stage, the excited particles come to the down state. If the same energy is reflected from the particle, then there's no Raman effect. But if the energy is different from the incident light radiation or the incident frequency or the incident energy, then it gives a different energy. It can be positive or negative. It is called stroke effect or anti-stroke effect. That is the Raman shift. How the energy shifted from the incident energy, it is the Raman shift. And Raman shift is characteristic to the chemical compound, and intensity of the peak is characteristic to the quantity. So using the Raman spectroscopy, we can quantify and identify the particles. And in the newer versions, it comes with the microscope. So we can look at our filter paper or the sample through microscope and project the laser beam to the area of interest or the particle of interest, and we can identify that particles. Or otherwise, we can, define the pixel size, and for each pixel, we can analyze the Raman spectroscopy data, and we can make a Raman imaging that illustrate the polymer type and the number of polymers, number of particles we can count. Like if it is kind of one polymer, we can count the green polymers that may be one compound, one polymer type. The red is one polymer type. So we can count the polymer number of particles as well as the type. And Raman spectroscopy, using the micro Raman spectrophotometers, we can analyze the particles smaller enough to like 10 micrometers. We can analyze the particle, reduce down to 10 micrometers. The micro FTIR, that is the, here we give the 400 to 4000 nanometer level wavelength energies. So that energy is used for the vibrations of the molecules. The vibrations of the molecules are specific to the specific bonds, and with that bonds we identify the particle type of polymer. The absorption, with the absorption of the particular energy, we identify the type of the polymer. And using the infrared, we can also use make the FTIR image mapping and identify the type and the number of particles. For these spectroscopic methods, sample treatment for sample dilution is recommended, and the lower detection level of the particles is little bit higher than the little bit higher in micro FTIR than in micro Raman spectroscopy. So I bit emphasize on the spectroscopic analysis because my personal view, it is when we do the environmental analysis, spectroscopic analysis gives much information on the sample distribution, size distribution, number of particles, and type of particles than the thermal analytical technique. So I emphasize on the spectroscopic analysis here. So in the spectroscopic analysis, we in-field polymer light breeze can be purchased, and we can compare our spectrum with the polymer light breeze, and heat quality index can be calculated. If the heat quality index is higher, the better the matching with the reference and our samples. However, machine learning with the personnel with chemical expertise are recommended for the better acquisition of data. The sample spectrum should be processed with background, baseline, and atmospheric corrections. The dilution, and there may be some, if we didn't treat the sample, prepare the sample, or digest the organic matter, there can be some interferences. This interferences gives the polyethylene versus fatty acids, because this is the Raman spectrum. This is polyethylene versus fatty acids, and this is for the nylon and protein spectrum. So prior digestion of samples will remove the interferences. So method validation and verification. What is method validation? So I want to talk a bit about the method validation. When we use the method, we want to verify, we want to confirm that method can be used to analyze our, we have to confirm whether this method is suitable for our analysis. That is done by the method validation. So for the method validation, we use repeatability. Same person analyze samples at the same time, and the reproducibility, analysis of different person and different times, and the sensitivity, the LOTs, we have to calculate them. So we do the recoveries also. For the verification, means that's a standard method. We have to practice as per the standard and see whether we can achieve that LODs. For example, for the microplastic analysis, the method may say that the limit of reporting is 3, but with the background interferences, we cannot report up to 3, then our level of reporting may be 10. So using each step, we have to verify whether we can use the method, and we have to define what are the LODs, LODs and other repeatability and reproducibility parameters what we can achieve in our laboratory conditions and our expertise. It is called the verification. So for verification of analytical data, reference materials with known polymer type, size, and number can be used for method validation. That is, we can get the reference materials, reference polymer types with known sizes, so that we can count the particles. So they gives the weight of a different number of samples. So we can add that weight, and we can see whether we can recover how much we can recover from the added sample. So it is recommended to record the spectrum correctly identify at least 4 types of microplastic at the claim size is lower limit of size. And recovery rate is calculated. It is number of beads after the filtration and the number of beads 5 into 100. If the recovery rates are higher than 70%, we can use that method in our laboratory as validated method. And there's another way that we can analyze whether we are doing accurately or not is to participate in proficiency testing or inter-laboratory comparison. Proficiency testing is there are some proficiency testing providers accredited under 17043 standard. They have high analytical facilities than us. So they prepare the sample, they test for the homogeneity and stability. They analyze the prepared sample for the homogeneity and stability, and they define a period whether we want the period. During that period, if we are participating for that proficiency testing, we have to analyze and report the results based according to their procedure, whether they will take the true value, the known value they get, or the population mean of the laboratories that participate for the program. They define the true value and they calculate its score based on the true value, and if it is less than plus or minus 2, we are in the population. We are not the outlier of the population and our accuracy is high. That is how we analyze the proficiency testing. Or we can do the interlaboratory comparison. We can prepare a sample, but we have to analyze with different laboratories. But here we have to assure the homogeneity and stability of the sample. And there's a PT provider for the microplastic analysis, VPAD, but they have conducted several PT rounds. But see, recently I couldn't find the proficiency testing program. So there's something to have discussed. It is the reporting levels. How we define the reporting level, RL. We can do a 10 blank sample and calculate the average of the 10 blank samples and add 3 times the standard deviation of these samples. It is our reporting level or limit of detection in some analysis. In spectroscopy, it is reporting level. So then we do our sample 1 and 2. We get the particle number 6, number of particles 6. But we cannot report this value as it is under our reporting level. We can report, this is 12. We can report this value without subtracting the background levels. Here, our daily bank of series is greater than reporting level. Our series is invalidate, so we have to repeat the analysis. So we cannot report any particle number or any value below our reporting level. So this is one part of method verification and method validation. We add 3% of standard deviation to allow to be confidently report to increase the confidence interval up to 95%. So this is there are thermoanalytical techniques. One is pyrolysis GCMS and the thermogrammetric FEIR. There's a pyrolysis in the GC. There's a pyrolysis oven. There we can insert the sample in an echo cup in an inert environment and known temperature in an inert or vacuum environment. Sample is pyrolyzed, decomposed in the absence of oxygen. Then the particles is fragmented in their weaker bonds and produce smaller molecules which are highly volatile. And once they goes with the carrier gas through the GC column, the compounds are separated and it can be identified in the chromatographs. In the thermogrammetric FGIR, the the sample is volatilized, and the time that gas is injected to FTIR, and the FTIR spectrums are taken. They are called thermogrammetric analysis. So, as conclusion, the message I have to give: only reliable identification at low numbers and low size, but time consuming through spectroscopic analysis. As microplastic antiques in laboratory environment, it is difficult to have lab blanks or reference unpolluted. So we have to do blanks for each and every steps to evaluate the contaminations. Standards will impose laboratories to verify and validate their method implemented. That new 2 standards is going to be published in 2025. One, the water with low organic matter content, the spectroscopic methods, and the thermogrammetric methods. They will address more quality control and quality assurance protocol and this in these gold standards. And still at the moment, no ISO standard, but there are some ASTM US standards for methods, and still no regulations. So we don't have the number of threshold for the microplastics in our drinking water. So those are to be developed in the future. However, to develop the regulations, we need standard methods, and the method should be standardized. At last, threat to our planet is the belief that someone else will save it. Let us each be the change, assuring the cleaner water and the future free of microplastic pollution. Thank you very much.
Thank you very much, Dr. Kushani, for your insightful presentations. We truly appreciate your contribution. Now, we would like to open the floor for discussion. Participants, please feel free to ask questions or share your thoughts.
Hello.
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 Yes. Yeah, I can hear you.
How much sample is needed to collect water for microplastic identification in groundwater? How much sample is needed?
As I talked earlier, that if we use the spectroscopic method, you can handle with large volume of samples, even though the particle number is high. So if they are clogging, you can take another filter paper, but it is recommended at least 1 cubic meter.
Okay.
Because
Hello,
if you are, yes.
Hello, madam. Very good evening.
Good evening.
I'm Dr. Dinesh calling from Hyderabad, India.
Yes, Dr. Dinesh.
Madam, basically, I belongs to pharmacy background.
Hmm.
The topic is very interesting, the microplastic, regarding microplastic. My question is only 2 questions, madam. Currently, in our day-to-day life, there is no human life without use of plastics. Our day start with toothbrush, which is made up of plastic. Our day end with toothbrush, that is also made up of plastics.
Yeah.
In such cases, how can we prevent the environmental pollution by minimizing the plastics? Because our life is completely combined with the plastics resources.
Yes.
And second question, madam, I'll complete, madam, please. Second question, what is the protection we have to take the industry, construction material emitting plastics? If any city like Hyderabad, city like Colombo, there is so many construction is happening. Even the construction material also emitting the microplastics, which is called as airborne plastics. So how can we minimize those things?
Dinesh, one option is we can go for the alternatives, like that in Central Environmental Authority of Sri Lanka has banned some plastic wrapping. They have used some the thinner gauge packings. They have restricted, and even the cotton buds they have restricted not to use the plastic rod for the cotton buds, and so many implementation, and it is with the circular economics. So we have to come up with the different options. Otherwise, we cannot reduce the use of plastic because it is cheaper and durable. So it is with the circular economics, it is with the different options. We can go for the reducing the plastic litter, like without using the shopping bags, we can use the paper or the cloth bags, but we for the if we are not taking those things, but if you want, we can. The other airborne plastic that is in the industries, it is possible with dust collectors and treating those collected dust, but the construction industry, that we have to look for the better options.
Okay, thank you, ma'am. Thanks a lot.
No, ma'am,
Yes.
can we collect the atmospheric microplastic in glass funnel as a sample? Can I use a glass funnel as a sample?
So that we use the white, not the glass fun, that we use the white bottles. But if you want to collect, if you want that, I use the glass white mouth glass bottle to collect the evaluate the environmental deposition during my sample transferring. However, that if I want to analyze the human exposure to microplastic dust, then some special devices that we can measure the human the respiratory intakes are available. In that case, we can use that filter.
Okay.
Yeah, for that NIOS recommended some
Hello.
pumps
Good
are
evening,
available.
ma'am.
Good evening. Yes.
Ma'am, I do have a question. Like in your presentation, you have mentioned about many detection techniques by which we can detect and quantify microplastics. So my question is basically that whether by means of any HPLC system can we detect microplastics?
That is not in literature, but the problem we have to then we have to dissolve the plastic material into the liquid system. But here the chromatographic system that the literature has used is the GC. They paralyzes the sample into a gas and analysis of gases. But those techniques I have not seen anywhere. But theoretically, we have to take the samples into a liquid form to inject into the HPLC.
Okay. Okay, ma'am. Thank you.
Hello, madam.
Hello.
Yes, madam. I have actually gone through some research papers regarding this microplastic. Several research papers are there, like absorption onto biochar, like that. So, based on that methods, they are using a specific types of microplastics, and their method is they are using some kind of methods like UV spectroscopy. So is it possible to use that UV spectroscopy in the analysis of this microplastic, a specific type of microplastic?
I have seen in 1 or 2 papers that they have used the UV spectroscopy also. for the microplastic analysis, but the basics really, even I too have to go through it.
Oh, okay.
But those methods are still very, I mean, use of such methods and the quality assurance are not properly documented. And as far as I know, that the spectroscopic and the GC the procedures will be recommended by the ISO standard in future.
If I'm to answer Udara's question about the UV using the UV method to analyze microplastics, it is just an indication. You can do that, but it is not a standard method. You can test with a particular microplastic type, but in the UV, since there can be scattering as well, and you can test it with a scanning procedure and get a peak in the UV spectra, but that is not really a standard method. And also when you have biochar, biochar also gives some particles to your solution. So you never know whether it is coming from biochar or it is from the microplastics itself. So it's not really good to use those kind of methods as standard methods. You will have a lot of questions from reviewers if you submit those kind of manuscript to papers.
Oh, okay, madam. Okay, thank you so much.
Dr. Kushani, you have many questions in your chat box.
Dr. Kushani?
Yes.
Yes. So are there any electrochemical or that kind?
Yes,
Electrochemical
there
methods
are
to
some
identify this
electrochemical methods to separation of particles.
Yeah, are there any sensors, portable devices, or such kind of devices to identify these microplastics?
For the separations, they have the researchers has used electrochemical methods for the separation.
Then
Only.
how can they separate it using electrochemical?
That I mean, they have introduced a charged and separated accordingly.
Electrostatic charge or?
Yeah, electrostatic charges.
Okay, thank you.
Ma'am,
Yes.
is there any methodology to calculate risk assessment from the contamination of microplastic from groundwater, yeah, from food samples like that?
Mm.
To Krishna, yes, there are many risk assessment techniques, but those are just calculated techniques and estimations kind of. It is not really an assessment. They are estimations that you can, you can assess how much of microplastics that can be exposed by a certain or an adult or a child, and then based on that exposure, as exposure amount or the abundance, that you can calculate the risk. So there are few techniques, but those are just estimations because we never know how many exactly is going into our body using water, groundwater, or food.
Yes, ma'am.
So they are estimations that you can
Yes.
do estimations for risk assessments based on these exposure assessment calculations.
Dr. Kushari, there are
Thank
questions
you, ma'am.
in the chat box.
Chat box, yes.
One.
One is how can do the air blanks in the lab? We normally open a wide mouth glass bottles and until we do the sample preparations and wash this bottle with RO water and treated as same the way as the samples and do air blanks. And we use the ARC software for the automated counting. It is recommended by CFAS UK, and for the optimization that we have that the intensity of the red, blue, and green light that we have to adjust accordingly. That is how we do the automated counting.
Can we use DI water instead of RO water? There's a concern here as well.
Yeah, you should do a blank for the water. When we use that plastic wash bottles, there's a contamination, at least 3 particles per liter. So you can use the distilled water blank. And each and every solution should be filtered with the same size filter papers before the analysis. I mean, so once after sample preparation is done, we can filter. Then on the filter paper, the particles are retained.
In case of analyzing PET microplastics in commercially available bottled drinking water, what will be the sampling method?
That we employ the same sampling method was specified in SLS 894. Based on the production, we have to collect the number of bottles statistically. If they produce 100 bottles, then we have to collect 3 like that. According to the standard 894 standard, we collect the number of bottles.
Okay, question from Ishani. When it comes to soil media, soil particles attached to microplastics, she's having a problem of getting absorbance peaks in FTIR due to the interference of soil. So we would like to get that.
So normally analyzing the soil particles, first the density separation is done, then the organic matter is digested, then the bound particle can be separated.
Can you suggest more promising methods to detect microplastics in edible fishes?
Edible fish, that I'm not sure whether spectroscopy or pyrolysis method will work, but in the pyrolysis, thermoanalytical pyrolysis GC, that we can, there as we detect the mass fraction, we can homogenize a large number of sample portion and take a representative one. But for the spectroscopic analysis, as we are counting the number, we cannot use energy to homogenize the samples, because then if the sample is broken, it will give the false results. So in that sense, the GC pyrolysis will be fine enough, but it depends on but in the fish flesh, the particle size should be very less. But so we have to whether that from that sample, once we homogenize the sample, the amount sample should be depend on the limit of detection of pyrolysis GC.
There's another question. What is the best method to polymer identifications of soil microplastics?
Whether it is soil or sediments, we have to separate it from soil and collect the microplastic first. The digestion and the density separation we have to do first before the analysis. So once we separated it from the soil, we can go for a spectroscopy as well as I think the spectroscopy will work better.
More questions if you have any.
Madam Dinesh again, please.
Yes, Dinesh, go ahead.
Madam, is this biodegradable plastic is a suitable alternative for plastics to prevent the contaminations?
Hmm.
Hello?
Yes, I think it will depend on it will depend on the risk analysis. Risk to us. For a certain amount, it will be degrade, but for the certain amount of time, it will be persistent in the environment. So the
But here, madam,
toxicity.
as a pharmacy graduates, we are using a biodegradable polymers for our drug delivery systems, control drug delivery, sustained drug delivery, or parental drug delivery, whatever the drug delivery systems, we are using a biodegradable polymers. Does it cause any environmental pollution? That is my question.
To answer Dinesh question, biodegradable plastics are, it depends on actually the use, as you mentioned, it depends on what you are particularly using it and what type of biodegradable plastic that you are using. There are some biodegradable plastics that are similar to the non-biodegradable plastic because they have been using the same monomers, but they are the plasticizers are different. And there are some biodegradable plastics, they are totally biodegradable. They are being produced by starch and the cellulose and these kind of things. So it depends on the source or the make of the biodegradable plastic. So depending on that, you have to be careful with the biodegradability. So if the degradability is high, and they have a background of petrochemicals, then you will have a problem. If it is produced by starch and cellulose like biodegradable stuff, then you don't have any issue. So it depends on the.
Yeah, we are mainly focusing on potato starch.
Yeah, then you don't have any issue because it is starch, then it is being degraded totally to sugar, so you don't have any issue.
So it depends on the molecular weights, as you said. Molecular weights of the polymers.
Yeah, monomer, including monomer types.
Oh, yeah. Okay. Thank you, madam. Thanks a lot. I have no questions now, please.
Okay. Are there any questions to be answers from chat window? I think I have raised all things here as well. Yeah, one more, Dr. Kushani. Madam, when analyzing microplastics in different macroalgal species, when the organic matter digestion with chemicals, it affects the different physical properties of the species. So different levels of digestion chemicals has to be used completely digest the sample. Isn't that going to affect the microplastic content?
Yeah, that digestion may affect the microplastic content, the shapes and color and everything. But then you can what we do when we are modifying the digestion solution, we add microplastic and we monitor the degradation time to time. That morphology we can analyze before the spiking, and after the digestion we can do a recovery test, as well as the morphologically we can analyze what the damages that has been occurred, the changes what has happened within the recovery tests. Recovery as well as the morphological analysis we can analyze when we are changing the digestion solutions.
Yeah, one more. Can you suggest some best digestion methods, chemical compositions to extract microplastics from biota, fish, prawns?
For the fish prawns, the alkaline digestion with hydrogen peroxide is recommended, but we do like that. The for 8 KOH and the hydrogen peroxide, 30% hydrogen peroxide, and the CFAS recommended the same solution like 30% hydrogen peroxide and sodium hypochlorite, but at our laboratory we use hydrogen peroxide and KOH. For the animal cell digestion, the challenge is the plant cell digestion because of the cell wall. So it needs more alkali content to digest the plant cells than the animal cell.
Yeah, that's all, I guess. Yes. One more question. Can we go for enzymatic digestion when digesting plant cells?
We can go for the enzymatic digestion. It causes less effect on the microplastic, but enzymatic, it is selective. Each enzymes are selective. So maybe we need trypsin, pepsin combination, or it should be tested which enzymes totally digest our sample. The enzymes can be selective to the several proteins or so on. And it is very temperature sensitive. We can pay much attention on that. And enzymes are selective, but it causes less damage to the plastic particles.
Yeah, there are many requests asking the recording files of this session. Yes, we can provide, and it will be uploaded by Young Scientist Forum, NASDAQ. So it will be available soon. Dr. Kushan, there's one more question as well. What would be the best method for the microplastic extractions from plants?
As what I told earlier, the plant cell digestion is much challenging because of the cell wall. So we may have to use depend on the more hydrogen peroxide plus more alkali solutions, or the Fenton reagents. Fenton reagent, the hydrogen peroxide and iron content may be changed. We have to change the ratios to digest the plant cells. I think the both digestion can be used. Some researchers I have seen, they have used the urea solutions also for the plant cell digestions. But we have to take care. We have to carefully look whether our plastic particles are digested with when we change the solutions.
More questions? Yeah. Since landfill leachate contains too many organic matter content, can we use the same alkaline digestion method or concentrations, or any suggestions to increase the concentrations?
I think the same 30% hydrogen peroxide and sodium hypochlorite, that what I normally use at the laboratory, will work. But we have to test, and I also believe that Fenton reagent will also work with the landfill leachates.
Okay. So no further questions from the participants. So as we draw these inspiring sessions to the end, on behalf of the Young Scientist Forum and Ecosphere Resilient Research Center, I would like to take this opportunity to extend our gratitude to everyone who made today's event a success. First, I would like to thank our resource person, Dr. Kushani Mahatantila, for sharing your expertise and valuable insights with us. So actually, your explanations has not only enriched our understanding, but also inspired all of us to think more critically and creatively about addressing all these environmental challenges. Once again, thank you so much, Dr. Kushani. And I would also like to express our gratitude to Professor Methika Vittanage, Founding Director of Ecosphere Resilient Research Center, for actively participating and engaging and giving us more contribution to this attractive session. And further, I would like to give our appreciation to Director of National Science and Technology Commission for your support and encouragement for initiatives like these webinars. And a special thanks goes to President of the Young Scientist Forum, Dr. Upadit Lenarachi, and all the steering committee members for their initiatives, leaderships, and all the facilitation process. Further, I would like to thank and give our appreciation to Ms. Tilini for her coordination and behind the scenes efforts to ensure the smooth functioning of today's event. Lastly, I would like to thank all our participants for your involvements. Actually, this one was very successful with your active engagement. Your presence and active participations are what make events like this truly meaningful. We look forward to seeing you at our future events as well. Thank you very much.